Okay, so my last video, I went through the cell cycle, which is the life cycle of a cell, and what it goes through as it grows and then divides to form new cells. Remember, we have the G1 phase, which is growth, the S phase, which we're copying the DNA so that when it create new cells, it will assure that each new cell has a full set of DNA. Then we have the G2 phase where we're just preparing to divide, and then we have mitosis or the M phase. So this video, I'm going to go through this M phase or mitosis phase and the steps of it. Remember, we use mitosis for a bunch of different things. Asexual reproduction, like a bacteria will just divide itself to form two new cells. It produces somatic cells or diploid cells, which have two sets of chromosomes, which means we can use mitosis for repairing structures in the body, healing wounds, and also for growth. Okay, so how does mitosis work? So remember this little acronym PMAT, PMAT. PMAT stands for prophase, metaphase, anaphase, and telophase. Now, one thing to remember or to keep in mind as you're going through this, you do have to know the phases. You don't have to know exactly what's going on. So I'm not going to ask, hey, when did the nucleus break down or whatever. But you do have to know the phases. So let's go through this. So big thing to remember is before any cell divides, the DNA has to copy itself and we form these sister chromatids. Remember, a sister chromatid is like the X structure. Okay. So in prophase, which is our first part, and again, this happened after interphase. So our first phase of mitosis is prophase. Basically, in prophase is when we start to see the chromosomes as sister chromatids, and the nucleus just kind of disappears. All right. And what we have is these little things called spindle fibers. You can see these are spindle fibers here, and you can see the little gold things. Actually, let me get my little pointer. Okay. You see the spindle fibers here and on either side, those little gold things. Those are going to reach out and attach to each of the chromosomes or each of the sister chromatids, and they're going to attach at those things holding them together, the centromere. All right. So prophase, the nuclear envelope breaks down, the nucleus goes away, spindle fibers attach to the chromosomes. In metaphase, these spindle fibers are going to move each of the chromosomes or sister chromatids to the center of the cell. So this is actually really easy part to remember for me when I was studying it, because metaphase, I just remember M is middle. So all of the sister chromatids line up in the middle of the cell. And they actually call this the metaphase plate. So this metaphase plate, the center of the cell where all the chromosomes are lining up. All right. So prophase, the nuclear envelope disappears. We attach them to spindles. In metaphase, these spindles move all of the chromosomes to the center of the cell. Anaphase, remember PMAT, anaphase, we're going to start yanking and pulling apart these sister chromatids. So now these spindle fibers, which are the gold things, are pulling each, like basically the half of each chromosome to opposite ends of the cell. Now, because we copied the DNA, okay, because we copied the DNA before cell division, as we're pulling those things to either ends of the cell, we're basically pulling a full set of DNA to each side of the cell. Okay, so you can think of anaphase kind of like a game of tug of war. There's a person down here and a person down here. And by the way, these things are called centrioles. These little I don't know, little gold structures right there. The centrioles are pulling these sister chromatids to opposite ends of the cell. So half going this way, half going this way, okay, going down. And then lastly, we have telophase and cytokinesis. Telophase and cytokinesis. Now, this last stage, so what has happened is we've pulled a full set of chromosomes to one end of the cell, a full set of chromosomes to the other end of the cell. Okay. Now, if we don't go through this process of cytokinesis, we would just have this big, oblong, weird-looking cell. So cytokinesis, the job of it is to divide the cytoplasm. So you see how we are kind of pinching this, kind of looks like the shape of an 8 right now. That is supposed to represent how the cytoplasm is dividing in half and kind of splitting right down the middle. Okay, this is called a cleavage furrow. This cleavage furrow is dividing the cytoplasm and giving half of the cytoplasm to what is going to be one cell up here, and half of the cytoplasm, which is going to be one of these cells down here. Also, in this phase, we are starting to reform two nuclei. Okay, and these chromosomes, you see how it says the chromosomes are relaxing. Okay, so now each of these new cells is going to go into interphase. So the chromosomes are going to break down and they're going to become chromatin again. And the nucleus is going to form, and basically the whole cycle starts all over. All right. Let me go back to this one more time. So telophase and cytokinesis. Telophase, the nucleus is reforming on both ends of the cell. And I'm going to cytokinesis to split the cytoplasm. If this was a plant cell, plant cell looks a little bit different, okay? Because right down the middle, instead of this cleavage furrow like we talked a minute ago, where it's kind of splitting and forming this figure 8 looking thing, in a plant cell, we get this thing called a cell plate that forms right in the middle to divide those cells in half. And the reason we have that cell plate forming is because plant cells have something called a cell wall. So they end up looking more block-like, and they're going to stay together, whereas these cells may travel to different areas. Okay, so let me go back one more time. I'm going to go through it again. So first of all, remember that we have the cell cycle. G1, this is just a cell living its life, it's growing. The S phase, it copies its DNA. The G2 phase, it's preparing for mitosis. And then we get in the M phase. And you remember, we've talked a lot about what we use mitosis for. So we start with prophase. Prophase, nuclear envelope breaks down, and the sister chromatids, because we've copied them, have attached to spindles. Metaphase, those spindles move each of the chromosomes to the center of the cell. We call that the metaphase plate. Anaphase, those sister chromatids get pulled apart to opposite ends of the cell. And all these little blue things is a full complement or full set of DNA going to opposite ends of the cell. Telophase and cytokinesis, I'm reforming the the nucleus on either end of the cell, and those chromosomes are breaking back down, reforming chromatin. And cytokinesis is splitting the cytoplasm. So we're going to form two new identical daughter cells, is what this is called, identical daughter cells. And then finally, just the big difference between plants and animals. Plants are going to form something called a cell plate to divide these cells in half, and that is mitosis.